Welcome everyone to Tofu Tech Talk. This is a little STEM show that I do where I capture Twitch streamers with STEM backgrounds and I force them to talk about super smart stuff so that the chat IQ will raise. So everyone coming in, I hope that you wish a lovely welcome to Slightly Caffeinated, our newest guest here. Oh no, the camera redid itself. I'll fix it. 2 seconds. I fixed the camera. All good. Now I'm Slightly
Okay, I was
Caffeinated.
like,
Would you like
it's like so.
Would you like to introduce yourself, please, to my community?
Of course. Hi, everyone. I'm Slightly Caffeinated. You can call me Caff because Slightly Caffeinated is too long. So I'm currently a third-year PhD student. I'm rising into my fourth year. I study tumor-immune interactions, specifically natural killer cells and their role in cancer and how they control cancer. And before that, I have some background. So I did my bachelor's in microbiology, which was very fun. Microbiology is just a wonderful, broad topic. And that ultimately actually piqued my interest in immunology. And then through some little adventures in between, I got interested in cancer, and I had heard about cancer immunotherapy and found that there is this really cool way where you can use the immune system to target cancer cells without having to rely on things such as chemotherapy, which we know are very, very stressful, like very toxic treatments. Oh yes, how is the audio? Is the audio okay? I just started going.
No, that's perfect. I always write it in chat so that my mods can check it live for me as well, because to me it's good, but I just want to make sure that everyone hearing, everyone is giving a thumbs up, so it's very, very good. So then you're doing a PhD in immunotherapy.
Yeah. So I guess more specifically, it's technically an immunology graduate program, and the lab that I'm in studies different ways that we can reprogram or retarget different immune cells in our body towards cancer. And one of these immune cells is a cell called the natural killer cell. So we have several different types or classes of immune cells. We have our adaptive immune cells, which are like T cells. I think most people have heard of T cells. And B cells, for example. And then you have your innate immunity, and these are typically immune cells that will respond very quickly. And that's where the natural killer cells sort of hang out and live. So we are interested in them because they don't require so much priming and activation as the T cells do. And they actually were discovered for their ability to kill cancer very easily and very quickly.
Interesting.
Yeah.
So okay, I'm gonna have to go back a lot because I didn't understand a lot. I'm so sorry. I'm not the brightest bulb in the shed, because as soon as you started talking about T cells, my Resident Evil ears picked up and I was like, oh no, is that T virus we're talking about? You see, this is why biologists are dangerous. This is where my conspiracy theory comes in. Anyways, you talked about T cells. Could you explain what that T cell is?
Yes. So a T cell is an immune cell. It's derived from a stem cell progenitor that lives in your bone marrow. And then it goes through this long development process in your thymus. During its time in the thymus, this T cell is being taught to, basically, if the T cell responds to any self-protein, it's killed, because you don't want your T cells to kill any of your own proteins, because that leads to organ damage, tissue damage, and it's very bad. So those cells are all killed. And then the T cells that do not react to any self-proteins, those are allowed to go out into your circulatory system, and then they do various functions such as responding to pathogens, viruses, bacteria. They respond to foreign proteins, so like cancers, because they have a lot of mutated proteins. And the T cells do this with a fun little receptor called the T cell receptor. It has 2 little like alpha beta chains, and there's a broad, like very wide variation in these T cell receptors. So they can recognize just hundreds, if not like thousands of different types of protein combinations. And that is how we have our immune system is able to recognize so many different potential viruses and bacteria and whatnot. And the cool thing is that your T cells, once they have, let's say you have, I don't know, a COVID-19 infection, and you have T cells that are specific for the virus, they get activated and then they'll clonally expand. So you'll get one T cell perhaps that it has the T cell receptor that recognizes the COVID-19 virus. It gets activated, it makes many copies of itself. And then these copies, after the virus has been cleared, they turn into what we call memory T cells. And your memory T cells can stay with you throughout your entire lifetime.
So this is how basic vaccines work as well then? The T cells, they learn how to battle that virus or that whatever it is, and then it has that memory of how it beat it last time, and it's ready to reproduce those cells if needed.
Yes. Okay. And usually the memory, it's just that it has the appropriate T cell receptor, and if it gets reactivated a second or third time, those cells, there's already a little population of them, so they'll expand much faster. So the second or third time that you're infected with something, the immune response will be much quicker, which is what you want to really wipe out a viral or bacterial infection. Yeah.
Just to kind of keep going on this train of thought, does that mean that the more you get infected with the same disease, the better you battle it? Or is that a fair statement or no?
I think that, so yes, I believe so. You're, like if you're getting, so I often think of like the flu, but unfortunately there's different variations of the flu. Yeah, different strains. So the target is always going to be slightly different, which is why you have to get your flu vaccine every year, because otherwise, the little pool of memory T cells and B cells, they're going to be primed for last year's version of the flu, not this year's. But if you do get repeated infections, I think it does help. But at some point, there's sort of like a limit where you're not really getting any more returns. Yeah.
Okay, I see. Really interesting. And then, so I asked about the T cells because then you went on to say some things about killer cells, right? Was it killer cells you called them?
Yes. Yes. Natural killer cells.
Okay, and what are natural killer cells built after we mentioned what T cells are? What is the difference and what are they?
So natural killer cells, they have a very similar function as T cells. So one thing I forgot to mention is, so your T cells, you have two different sort of main classes. You have your CD8 T cells, and then you have your CD4 T cells. And the CD8 T cells are typically the ones that are focused on for cancer immunotherapy because they can directly form a little like immune synapse. So it's like a little tunnel or connection with tumor cells. And then they inject cytotoxic granules from the T cell into the tumor cell, and this causes the tumor cell to die. So there's this very direct killing mechanism. They can also kill through other different ways, but NK cells or natural killer cells, they can kill the same way as T cells, where they form this little synapse, they insert their little cytotoxic granules, and then the tumor cell dies. So the killing mechanism is similar. However, the recognition is very different. So as I mentioned, T cells have a T cell receptor that they use to recognize proteins on the surface of cells all through our body. And these proteins, they're presented on something called MHC1 in mice, major histocompatibility complex. Or in humans, it's called HLA, or human leukocyte antigen. And so if you have ever gotten we all have our own unique HLA type, and different mouse strains have their own MHC type. I know I mentioned mice.
I don't know if you know, our community is called lab rats, and we've had a few biology people recently, and a lot of people experiment on mice, so a lot of my community might be tucking their tail at the mention of this.
the little test subjects. So yes, so going back, so T cells, they recognize the proteins that are presented on MHC1, and then there's also some other activation receptors that are required for the T cell to get fully activated and then actually kill that cell. Obviously, you need lots of checks and balances to make sure that your T cells are not just killing, normal cells. NK cells, on the other hand, they don't have a T cell receptor, and their development is completely different. They don't develop in the thymus. However, what they do is, so they have so they have two different types of receptors. They have receptors that recognize activating ligands, and then they have some receptors that recognize sort of inhibitory ligands. And typically what happens is if your cell is healthy, it has the MHC1 on its surface, which is presenting protein. And if the protein is self, your T cell will not kill it. Now, if for some reason that cell loses that MHC1, usually when cells get infected with virus or they become cancerous, one of the escape mechanisms is for that cell to reduce or completely lose expression of MHC1. So now the T cell can't see it.
Oh, it can't detect it.
But
okay,
it can't detect it. It's like taking your name tag off and just throwing it away, and like going rogue. But at this point, the natural killer cell, which is usually inhibited by MHC1, it's now free to go ahead and kill that target cell because the inhibitory signal has been removed. And so now that natural killer cell is going to kill that cell. There are, of course, other little nuances, like are there enough activating ligands present on the cell? Ligands are just like proteins. So you could imagine a cell and it's studded with different proteins. Cells are not these smooth little balls. They are actually very they're kind of squishy shaped. It depends on the cell type, of course, but they're not perfectly smooth. They have a whole bunch of different proteins and different lipids and sugars on the surface. So they're very bumpy. And when they get stressed, for example, from like a viral infection or from cancer, they're going to sometimes produce certain proteins as a response to that infection. And then those proteins are recognized by the natural killer cells, and they can just go in and immediately kill the cell. They don't need prior activation. They don't need clonal expansion like the T cells. So natural killer cells we kind of think of as sort of first responders to any type of bad
more like
cell
assassins
in your body. They're definitely very cool assassins. I would say the CD8 T cells and the natural killer cells are the assassins of the body. Natural killer cells are just much more, they don't need that priming to get going.
And they produce naturally in the body? I don't know if you mentioned this and I didn't catch it.
Yes.
Okay, where?
So
Sorry.
They also originate from the bone marrow, and then they go through and they develop into various different subsets and classes with different functions. Most of your immune cells come from your bone marrow, which is why our bone marrow is so important. And that's why sometimes when people have an issue with their immune population, they'll get a bone marrow transplant from someone with a healthy set of stem cells that will turn into healthy immune cells. Okay.
And so your research is on these killer cells?
Yes. So I'm interested actually in how cancers evolve and mutate to avoid killing by natural killer cells. And I guess I should go into a little background about why that's important. So currently for immunotherapy, we use a lot of T cell focused immunotherapies. So you might have heard of CAR T cells. These are T cells that are engineered to recognize specific proteins that are only found on the cancer cells. And these work great for blood tumors. There's actually several that are approved by the FDA for like leukemia, lymphoma, and they work fantastic for any hematological or blood cancer. However, these CAR T cells, they're not as effective for solid tumors. What happens in solid tumors is that the tumors, as they grow, they get mutations. And these mutations are usually, it's pretty random. We do think of, so there are certain like driver mutations, and these mutations are initially, you could imagine the very first cell that became a cancer cell, that mutation, which is typically we see these mutations in genes that regulate the cell cycle. So a cancer cell is a cell that no longer follows the rules of normal cell cycle. It's rapidly dividing, and oftentimes it's very bad at repairing its own DNA damage. And so as it's dividing, it's copying its DNA and it's making mistakes in its DNA replication, and this results in random mutations. And if those mutations are beneficial to the cancer cell, then they will be selected for, and you'll get a tumor that kind of has maybe different subsets of cells that have all different types of these mutations. And usually the most selectively advantageous mutations will be the most represented. And what can happen is the mutations can make these cancer cells sort of invisible to the T cells by loss of MHC1 or HLA. So the T cells can no longer recognize them. And sometimes another way, so the CAR T cells, they kind of bypass that because they don't need the MHC1 recognition, because they actually have a special receptor that can recognize any protein on the surface of the cancer cell. But you could imagine if you have this CAR T cell just coming in and killing off a lot of these cells, you'll eventually have some cancer cells that have mutations where they've lost that protein, and they're now invisible to the CAR T cell. And so we're interested in developing alternative immunotherapies where we use this different class of immune cell, the natural killer cell, because they don't rely on a lot of the same recognition processes that T cells do. So, yeah.
Okay. And that's your focus. So you're specifically working on research for helping people with cancer. It's not even like just observing what cancer does or what the natural killers do. It's instead you're actually actively developing something. Would that be a fair statement?
A little. So it actually is a little bit more basic science than that. So we do have a therapy that activates natural killer cells. So there's certain signaling pathways in different immune cells so there's one called, I don't know if I should get into this too much, but there's a signaling pathway called the cGAS sting pathway. All you need to know about it is that this pathway senses broken bits of DNA in the cell, and then it spits out these inflammatory cytokines. Cytokines are these little proteins that will activate all of your good immune cells and some bad immune cells, which we'll get into maybe, but they'll activate your T cells and your natural killer cells. And when the cGAS sting pathway produces these little cytokines, you get a huge influx and activation of the natural killer cells to the cancer. And so we're interested in using therapies that sort of bypass some of the traditional steps of the cGAS sting pathway and make it more activated. So we get more of these cytokines and therefore more activated natural killer cells. And this works really well in different models of cancer in mice. And actually a lot of biotech companies have been trying to develop this for humans. There was a couple companies like Aduro Biotech where they tried to do, they ran a couple clinical trials with this sting agonist, but it did not work in humans the same way it worked in mice. So one of my projects that's kind of a mini project right now is trying to understand, like, is there some delivery issue? Like, is the sting agonist not getting into the cell appropriately, or is there some difference between human and mouse sting signaling with this agonist that we just weren't aware of? So that was kind of, to be quite honest, it's on the back burner right now because there were some technical difficulties. And one of the big things when you're a PhD student is you want to graduate. You want to graduate soon. So my advisor was saying, like, oh, let's also have a secondary project that's safer, where you can actually know you'll get a paper from this.
Actually,
like,
get a
cool,
PhD out of this. What year are you again?
So, I'm third going on fourth. Yeah.
No, this is the last stretch, so you do need a lot of more work.
Oh, it's actually so it's actually a halfway point for my program. A
No.
lot it I know that PhDs are shorter for people in Europe and Ireland and Australia, typically 3 years, 3 or 4 years maybe. Um, Here in our program lasts about 6 years,
Okay.
sometimes 7. In
Oh
our
wow.
department specifically, um, people are just trying to do really hard things with mice, and mouse breeding takes a very long time. And so
Mm,
to do your experiments, you have to wait months
mm,
sometimes to just have the mice to do the experiment. And then what if it doesn't work and you have to go back to the drawing board and start over?
yeah.
Um, so it really is it's a long stretch, and so I'm trying to just pace myself and get through it. So I consider myself at the halfway point. My advisor is a little bit more positive than me. He's like, oh, you'll be done in 5.5 years. But we'll see about it.
Yeah, no, I think it is true
So,
about depends on the department. I do know that in my university, the couple of biology human beings that I've spoken to, because for some reason those departments don't really mix with physics, they do tend to be a little bit shorter. It's not uncommon for me to hear people finish in 3 years, but in physics,
mhm,
unfortunately, it is very long in my department. So I was on the 5 year or 4.5 year mark, but pretty much 5 years,
mhm.
and that was pretty good. That was pretty good. We do have a world
Yeah.
record in our department of someone finishing in 3.5 years. That was the record in history.
Wow. Wow.
But that, yeah,
That's
it
pretty
is
amazing.
very, very long. Yeah, and I think it's just kind of
Yeah.
this kind of safety net. They want to make sure that you have a really well-rounded PhD. But it's nice that your supervisor's actively, going out of their way to ensure that you're getting a doctorate out of this. Because look, you can do all the research you can, but ultimately you're there for a reason as well.
Absolutely. Yes. I think it's very important to pick a good advisor for grad school. And I don't want to switch too far away from like the sciencey stuff. We can definitely talk about picking grad school
Mm.
advisors for anyone who is interested. Um, Also, I actually wanted to say when you said physics people don't mix with immunology people, I think there is, so I know physics is different from biophysics, but we have a really fabulous biophysics department
Class.
at my school. And I actually was very interested early on, my first year of graduate school, of going into one of these biophysics related labs because we study, well, in those labs, they study how, like the tumor, like basically the tumor cells and the immune cells, there's these immune cells called macrophages, how their like actual physical interactions and like the stress and the force and like the mobility of the cell through different like rigid surfaces, how that affects the actual immune response. So there's this whole field that I think is still very new, where people are studying kind of the physics behind immune cells and cells of the body, and how those different structural changes convert into different biochemical signals in the cell, and then how
Mm.
that gets translated down to like the DNA level and what
Yeah.
proteins are produced and stuff like that.
This
But yeah,
must be
I
such
think
a cool
there's a
field,
mhm.
interdisciplinary
Yeah,
especially. And like I suppose it's
yes.
always cool to hear about biophysics in general,
Mhm.
just because I feel we always I don't know if anyone else has that feeling that biology and physics are kind of very, very far apart. It's chemistry that might bind them together, if that makes sense. There's an order to the sciences. It's either like physics, chemistry, biology in this line, where you can do interdisciplinary, or the
Yeah.
other way around. But it's very rare that we in reality talk a lot about biophysics, but it is a really cool, like almost like two different worlds coming together.
Yeah.
No,
Yeah,
absolutely.
that's very cool.
I have a few questions logged, so I will start
All
questioning you
right.
here, if that's okay. So we might end up going back on a lot of stuff that you talked about, and obviously a lot of people coming in. Welcome everyone in. We are just about like 20 minutes into Tofu Tech Talk. We just did an introduction, and now we're moving on to a few questions. So if you have any follow-up questions, make sure to log them with the pinned comment up on top of chat. Now, Paranoid is asking, why do T cells attack some invaders but not others? I think that question was logged before you explained with the ligands.
Mm, mhm.
But if you
Yes,
want
like
to just
ligands,
review it
ligands.
again. If you want to
Yeah,
review it
yeah.
again.
Yeah, so the T cell recognizes its invader using a T cell receptor. And I guess when I'm saying invader, we can think of viruses, bacteria, cancerous cells. So the T cell has this little T cell receptor, and it will recognize a specific protein that is bound to this molecule called MHC1, which is typically on like some other cell, for example. If it's a virus, for example, the virus will usually infect a cell, and then some of the viral proteins will get displayed on MHC1, and then the T cell will, so if it's the proper T cell clone, it will recognize that peptide and then go in and destroy it. So I guess, so your question was, why does the T cell recognize some foreign invade or some like viruses and some things, but not
Yes.
others?
Why some invaders but not others?
So I think it really depends on, I mean, like there is a lot of variety in the T cell receptor, but you can imagine that since it's only recognizing certain like combinations and segments of proteins, so the proteins, they are broken down into little peptides, and these, you can think of them as like take a protein, which is this glob of different amino acids, stick it through a paper shredder, and you get little peptide fragments. And so the T cell receptor can only recognize certain types of these peptide fragments. So you could imagine that if it there's a peptide fragment specific to a certain virus that your T cell receptors can't recognize, you're not going to get an immune response. And so yeah, so that it's a little game of fate a little bit. But yeah, I guess that's that's my explanation for that question.
No, perfect. That's perfect answer, especially since we did go back a little bit. I have a couple of more here. Is picking grad school advisors much like picking a starting Pokemon? Sorry, I misspoke. But does it feel like that? Did you have like 3 options and you had to choose between 3 supervisors?
Yes. Yeah. So, oh man. Well, first of all, the Pokemon analogy is very interesting. So the way you typically start off grad school is you'll do rotations for your first year. Typically people do 3, sometimes people do 4. Some people don't do rotations. They just know. Maybe they knew the professor before applying for graduate school and they just go straight in. Those are direct admits. And I highly recommend, if any of you are interested in going into grad school, look into direct admission, because it does bypass a lot of this unnecessary sort of rotation stuff. But I will say, before I say it's unnecessary, the rotation is very useful for getting a sense of the lab's environment, which is very important. So a lab is typically composed of the advisor, the professor, and then you have your lab managers, and then you have postdocs. These are people who have done their PhD and they're now doing their own separate research project to try to publish papers so that they can get a faculty position. And then you have grad students like me, and we're just like dootsing around in the lab trying to graduate. And then you also have undergraduates, and you want to find a lab where the sort of like the advisor is creating a community within the lab that is supportive, that doesn't promote people throwing other people under the bus. You want an advisor who is very open to facing conflict in the lab and solving that conflict. What I saw in some of my rotations was that there were some advisors who just ignored conflict amongst lab members. And this creates a very toxic environment to work in. And you might think, okay, well, I don't like dealing with drama. I'll just ignore the drama, and just do my PhD. But honestly, it will take a toll. It will take a toll on you. And it's better to just go into a lab where the advisor creates a very conversational and safe environment for people to work through their problems with each other. And then also it is helpful if everybody in the lab sort of likes each other. And so when I was picking my lab, one of the things that was very important to me was this thing, because I had had some pretty bad experiences in the past where I had been in some labs where people were a little like, the environment was just a little cliquey. So you'd have different cliques of people, and there was a lot of gossip, and I just didn't like it. It was just like such a drag to come in, on a Monday and, see people being cliquey and weird with each other. So I asked my advisor, how do you handle conflict? And his response was, I sit the two people down, I have a discussion with them openly and go from there. And then also just the people in his lab were very, it just felt like a second family to me. Like I had been adopted by a second family, and that was very important. And people are very open to sharing their resources, sharing their knowledge. And I ultimately picked the lab for that, as well as the research interest, which is the kind of obvious one. But I think sometimes people forget that people matter, and you want to pick a lab with people that you truly enjoy and like.
Yeah, your experience of getting into a PhD program is completely different to mine. So it's really unique and very important for people to hear this. I suppose, especially since I'm going to assume, especially speaking from maybe a USA perspective and a maybe even a field. This is mostly possibly biology focus as well. Again, it depends, but no, it's a completely different experience, so it's really good.
How actually was your admissions process? I don't actually know how they do it.
I'm pretty embarrassed to admit my one. My one was, I was graduating with a master's degree. I ended up, having very good grades, top of the class and all of that. And a few supervisors walked up to me and they were like, would you like to do a PhD program? I was like, yeah, sure. And then one of them came up to me and said, well, look, I got funding. Do you want to hop on this train? And I was like, cool. I didn't know what the research project was on. And yeah, that happened. It was very quick.
Awesome,
I just didn't know what to do with my graduation after my master's. I was like, I don't have a job lined up. And they were like, well, we have a PhD program here available already. Do you want to go on this? I was like, yeah, sure, why not? So yeah, it was an accidental PhD. It was a side quest, exactly. So ended up sacrificing 5 years of my life for it.
I mean, it's a good side quest. I think ultimately at the end you come out with a lot of skills, and also just a character development, right? That's what side quests do. They build your character. Yeah.
lore building, exactly.
Yeah.
We have another question from Polaroid. I think this is the last logged question, but I have a whole bunch of my own, so don't worry. Is it possible to accurately stimulate cell behavior in machines, or is it too complex? So this is a little bit outside what we were talking about, but you are probably one of the better people for us to ask this question, since you do a lot of research and work in this. So
stimulating... Wait, let me understand. So is it possible to stimulate cells in machines, or...
Cell behavior in machines.
Cell behavior in machines. I don't quite know what machines, like what is an example of this?
Oh, simulate. No, it's stimulate. The question said stimulate. Maybe the question is simulate, but it says stimulate, so cell behavior, which are two different words.
Oh, simulate. Okay. No, you're good, paranoid. Is it possible to simulate cell behavior, maybe like using an algorithm? So yes, my computational background is very weak, but I do know that people can develop softwares that simulate the development, and also the development of different cells. For example, this is all based off of real biological data that they've already collected, but then they can sort of they use this sort of time scale that isn't, it's a proxy to true biological time scale, so like what we actually experience, but they can show how, for example, a T cell develops in this set time scale that they've used in their software. Again, I am probably not the person to speak about this. Now, in terms of stimulating, that's actually, I don't actually know of any examples of people trying to model stimulating. Oh, wait, that's the wrong question.
Yeah, we corrected it that it was simulating. So all good.
Sorry. The other thing I've seen people do, though, with different programs is they will look at how they'll try to model how a tumor might evolve over time with different selections used. I don't know how biologically relevant that is in some cases, because it's a little bit more exploratory, and I don't know, like I don't see a lot of people using that necessarily in my field, but it is something you can do.
Okay, I see.
Yeah.
I suppose to round up the whole discussion, finally, so we were talking about, the natural killer cells that you were saying. You explained the T cells and why the K cells are better, and why you're focusing on that research. I suppose I know that you are still a PhD student. A lot of your work isn't published, but is there any kind of like conclusive statement or story you can tell about your research? Did you discover anything so far that you're allowed to share? Because I know, obviously, you might not be allowed to.
Yeah. Let's see. So one thing that I have seen is that when you use our STING agonist therapy, it does elicit a very strong like natural killer cell response that does in situations. So again, I have some contrived mouse models where I will knock out certain genes, and then I'll look at the response to T and NK cells. So for example, if I knock out a gene, it will make the cancer resistant to T cells. But then my question is, can the natural killer cell that is stimulated and activated by my therapy, can it kill this escaped tumor? And I have seen that it works quite well, and I'm able to model this similarly to what we see in actual patient samples. And so yes, I think like I'm able to I have a good model working. I think ultimately though, the more interesting part of my project, which is understanding more novel mutations that will make these tumors resistant to the natural killer cells, that is still very much in progress. And I have a lot planned and a lot on the table that I'm prepping for. So I don't have anything exciting yet in that regard, but my models are working pretty well, and our therapy is working pretty well. So yeah.
You better feed the mice extra treats so they'll be good. I
more
always
bananas.
I always increase my chocolate rations to my lab rats, from like 25 grams to 20 grams every now and then, and it sure excites them.
Yes.
They perform better.
Yeah.
So I suppose
Anyways.
to move on to a bit of more fun, because we are the scientific questions are slowing down a little bit. I have a few fun stuff to ask. First of all, as a scientist, as a biologist working in the lab. Okay. We have one question coming in, and I think my question is fun. So I'll stay on the science question, and then I'll move on to the fun question.
Yeah.
What do you think cancer treatment will look like in the future? Will advances in science and medicine ultimately make it easily treatable disease? That's actually a really nice question.
That's a good question, Commandant Herder. I might have said your name wrong. So the current status. So I guess to quickly answer your question, what we'll see for future cancer treatments is a lot more, I'm going to use this buzzword, precision medicine. And what this means is that we've gotten really good at, in the past few years in terms of technology, we've gotten really good at taking patient biopsies out of the patient that has the tumor, and then looking at the proteins that are being presented by their tumor cells. And then this tells us, okay, what kind of mutational signature does this tumor have? And how can we provide this person a personalized therapy regimen that's specific for their tumor? Because before, like the current way that we treat a lot of cancers is with radiation, which introduces a lot of DNA damage, and ultimately this overwhelms the cells before they can sort of mutate away, although they still do oftentimes. But you can kill them with radiation, or you can kill them with chemotherapy, which again also causes a lot of DNA damage. But as you know, it targets a lot of rapidly replicating cells, so you lose a lot of your immune cells, you lose your hair. Like, it's just awful, and it does horrible things to bone density, so people have more fractures. So what people are now looking at is, instead of using these like what I call sledgehammer therapeutics, we're looking at ways to design the therapy regimen for the patient specifically based on their mutational signature of their tumor. And then what you can do with this is you can either use certain chemical like metabolic sort of therapies, which I don't study, or you can use immune therapies that you can use, like the CAR T cell, as I mentioned, which you can target against certain proteins, or you can use, there's this new thing that's very interesting. There's these vaccines. Now, these are not vaccines that you take before you get cancer. They don't prevent cancer. These are vaccines that you give to a patient who already has the cancer. Oftentimes, the vaccine contains peptides, which are those chewed up proteins that are presented by that person's tumor already, and then this peptide vaccine is taken up by the dendritic cells in the patient, which are these little cells that present the peptides to your T cells. And so then you activate your T cells and you get a really strong response against the tumor because of this vaccine. So that's really nice because it's cheap to make. Typically, it's cheaper to make than CAR T cells. And it's what we call off the shelf. So you can already have it available before you even get the patient. So you can just treat them right away without there being a lag time. Because oftentimes, one of the reasons people do so poorly with therapy is because their treatment is started too late. So this personalized medicine is very important. Another thing that I see helping the field is detecting cancer early, and also being able to detect the cancer throughout treatment so that the physician can change the treatment as needed. So one way that a lot of companies are looking at is looking at the circulating tumor DNA in the bloodstream of the patient. So instead of doing invasive biopsies, they're taking a blood sample from the patient and they're able to sequence and find tumor DNA. And this tumor DNA they can use it as a proxy for how much of that tumor is still left. So if you get more tumor DNA, then typically the tumor is bigger. And so there's a lot of studies right now that are trying to validate this process. And they have found in several studies that the circulating tumor DNA was detected before the MRI and CT scans picked up the person's tumor that was coming back, because usually sometimes these technologies you'd completely miss the tumor if it's very small. And if someone's in remission, so their cancer has been sort of killed to the point where it's no longer detectable, you want to be able to know exactly when it's coming back so you can start therapy and then squish it
before it rises up before it arrives.
Yes.
Okay, so your kind of outlook of the immediate advances in science would be vaccines, not the preventative vaccines as in the peptide injection that you were talking about, and also detecting the cancer early via blood samples.
Yeah.
Okay, thank
you
so much.
There's a lot of other things, but it would take too long to get into all of them.
Of course, because we have loads of questions that were logged in. Agathor actually asked this earlier, and I'm so sorry, it was very rude of me. I missed it. It was one of the more immediate ones, and apologies. It did seem like a good question. Not that I understand any of the words here, but they sound fancy, so I'm going to pretend. I'm going to put on my smart glasses. What's your opinion then on ADCs? So, what would you say the immunotherapy route has more potential despite the ADC field growing rapidly?
I think, let's see. Oh my gosh, I'm forgetting this acronym. Are these the antibody? Okay, I'm going to be quite honest. I forgot what this acronym means.
Yeah, no worries.
ADC. So I'm going to Google it.
Antibody drug conjugates.
Yes. Okay. So I'm guessing that you mean... So what this means is that you can use an antibody that targets a protein on the cancer cell, and this antibody is also linked to a drug which can then be cytotoxic to that cancer cell. So you can target the cytotoxic molecule to the tumor cell, and then that'll kill the tumor cell and not any of your other cells. So I have not seen too much coming out in that field, but something that's very similar to this therapy is these bispecific T cell engager antibodies. And what this means is that you make an antibody that can bind to the tumor antigen, and it binds to an activating portion of the T cell. And it forms like a bridge, almost like holding hands. It pulls the T cell and the tumor cell into conjunction with each other, so they can form that nice little immune synapse, and then the T cell can kill the tumor cell. And also, the most important thing about this is that the T cell gets activated by the antibody without needing to be activated by like the dendritic cells and other prior activation. So it's a more, you're bypassing some of those activation steps. And I do see, like, especially in 2023, I was reading a review of the hottest immunotherapies for 2023, and bispecific T cell engagers were up there. There's a lot of companies working on that. So I think that's a really interesting field. And I'm actually testing one in my own studies as kind of a fun little extra collaboration, extra projects, you know.
Of course,
just to keep me busy.
We have totally plenty of time to be doing that. Thank you for the answer. That's really good. Agatha says that's so cool. I think they're also experienced in this field, if I'm not mistaken. So it was great to get a question like that. Another question is, if you could forward time, would the human body eventually develop the T or K cells to fight cancer by itself?
So your natural killer cells, the NK cells, they are actually always going throughout your body,
so
and
they're present
monitoring.
Right now in my body.
They're present. Yes, you already have natural killer cells. Okay. You have approximately 5 to 10% in your bloodstream, and it's very important that they're there, because actually, as we speak, you probably have some cells that are maybe mutated, transformed, and the natural killer cells are there to kill them right as they form. The reason cancer happens is because there's just so much of this cell replication that it overwhelms the initial T and natural killer cell responses. Now, if you were to so your T cells, they're always present. Your NK cells, they're always present. Fast forwarding time, I don't think it would necessarily change anything. Yeah, but yeah, your body. So the other fun little thing is that people have seen that when you are getting poor sleep or you're highly stressed, your natural killer cell activity decreases a lot, and you're much more prone to getting cancers and other issues because those cells are not out there actively patrolling. So get your sleep, get good sleep, and don't be too stressed, because cortisol is the enemy of the immune system.
I have a lot of lab rats to shit talk, because I know a lot of you insomniac people be staying up all night watching this stream or getting up early. So I'll be giving out a lot more these days now for anyone not getting enough sleep. Jesus Christ.
Yeah.
Be right back, going to bed. Perfect response in the chat. Absolutely appreciate that. Take care of your health, take care of your body.
Agathor makes the ADCs. Oh, that's very cool. I would be very interested in chatting with you about that sometime. Yeah, that's very cool.
Just to kind of throw out there, Agathor and everyone else, the way to follow Kath is that in the title of the stream, there is an @ link immediately, the first word, to Slightly Caffeinated's Twitch stream. But not only do you just need to click on that, hover over it and follow immediately without just leaving the stream, you can also follow all her socials. So if you want to catch any of our other content, make sure to do so. Just a shout out there. I'll do it again in the end. Just to make sure.
My socials. My socials are very, very inactive at this time.
I see.
I'm very bad with the socials.
No worries.
But you can still check them out. Especially, I'm trying to be better on Instagram, but I do try to stay off social media as a rule to myself. But it is, when I have inspiration, like a funny video idea, I'll put it on my Instagram.
Nice.
Or if I have a coffee that I'm proud of, I'll put it there. So thank you for that shout out.
Preserve has a follow-up question. Is that the same mechanism by which when we get run down, we are more prone to viral bacterial infections? Is that the same idea?
Yes, exactly. So when you are getting more stressed out or you're just not sleeping enough, this directly impacts your immune system. Even diet impacts your immune system, and there's a lot more papers coming out on that. But what can happen is if you're fasting, for example, so there's optimal levels of fasting. I'm not going to get into this too much, but if you're stressing your body out by not eating enough at certain times, your immune cells will respond by actually going back into the bone marrow and being sequestered there. And so they're not in the bloodstream, being present for potential viral or bacterial infections. And so that's kind of a bad thing. But yeah, and broken heart syndrome. Oh my goodness. So I am actually planning to do a little talk about broken heart syndrome tomorrow, which is so a fun little spoiler. What happens is when people get very, very stressed, usually older women, postmenopausal, this buildup of cortisol and other stress hormones, it can actually trigger this condition where the heart starts beating irregularly. It becomes a little like ballooned, and there seems to be some role of the immune system in this. Although, as I was reading the literature, it's very unclear still, like if it's causative or more of a secondary condition. So I don't think the immune system is causing this condition, but it does seem to perpetuate it. We're looking at immunotherapies as a potential treatment for people who are suffering from broken heart syndrome.
See, okay, we need to catch a Slightly Caffeinated stream live tomorrow for that. What time is that, by the way? Just asking for a friend.
So that'll be at 8 p.m. Pacific Standard Time. Yes.
That's like 2 a.m. my time. Like Jesus Christ.
Oh God. No, you better be asleep. You need to be asleep.
I can't. My body needs to fight cancer here, so we can't be staying up that late. Wait,
yes, no,
6 a.m. my time. Oh Jesus, that's even worse. Anyways, moving on. The Fumbling Scientist, which is a great username, by the way, asks, due to the nature of the chemotherapy and its interaction with the immune system, at what point do physicians start prescribing antibiotics? Is it more preventative if they treat it as it comes up?
So I think this is because you're thinking because the chemotherapy destroys a lot of the immune cells, you need to protect yourself preemptively against infection. That's why a lot of cancer patients, they're considered immunocompromised. And if there's someone in your family or friend group that has that's currently on a chemotherapy regimen, it's very important that you do not bring any flu or sickness to them, because their body will not be able to respond well. So for prescribing antibiotics, that exact timing, I'm not sure. But I know that it does happen on a case by case basis for patients. I don't think I understand that it's necessary to prevent infection, but I do hesitate a little to say that it's a good thing, because when you're prescribing antibiotics, you're also wiping out a lot of really important a lot of important good bacteria. And this can screw up your gut. And we're seeing more and more that the health of your gut is very important for the function of the rest of your body, including your brain. The gut-brain axis is very strong. So like if you're having, like issues in your stomach, for example, like different, like, so if people have an offset in, let's say they have more bad bacteria present in their gut, whatever that may mean. Again, this field is very, there's no prescriptive thing out there yet for your gut microbiome, because it is so hard to study. So we can say good or bad bacteria, but we don't necessarily know like what should we be doing then to like make our gut microbes better. Now, a lot of people will say eat more fermented food, and that'll improve like your overall mood and energy. Alcohol.
That's fermented fruit. Look, you said it, not me. Like chat, you heard it live on stream. Fermented fruit.
Yeah, so alcohol is not really a friend of your gut microbiome, unfortunately. But I still drink my alcohol from time to time. But yeah. Anyways, sorry, I don't have a more specific answer for your question.
That is good. I clustered the questions. Some were a bit more sciencey, and others were about grad school. So I kind of clustered these questions from 10 minutes ago. So one question was, outside of your research, do you have any other responsibility as a PhD student in a US university? So this is very specific to you, but it'll be nice to share your experience.
Yeah, so we have to do a lot of teaching. So when I say a lot, it's actually not that bad. We have to do 2 semesters of teaching, and most American PhD programs will require some teaching at some point. But there are some schools where they don't have an undergraduate population, so you don't have to teach. You might be required to teach graduate students in that case. But I just finished my teaching requirement. I'm free. I can now focus on research. But I will say, the teaching, I think it's a very good experience, and I think it's necessary because one, you become more of an expert in your field as you're teaching the subject, and two, you learn how to present material better to people versus, like, let's say you're just siloed in your lab and you're not really talking much with others, then you don't get an opportunity to like actually discuss your research, and it can just become very much like this little silo that you're trapped in. So it's very important to interface a lot with students, and I think the teaching requirement is necessary and great. I know a lot of people hate it. I didn't always enjoy it. It was very exhausting. We had a lot of pre-meds in our classes, and the pre-meds were very, very like nitpicky about their grades and their points. But there was still some
you don't
wonderful
get paid
students
enough
that I
to
really
deal with
enjoyed.
it. Yeah, no.
Yeah, but it was fun. I enjoyed it.
Nice.
But yeah, that's the main responsibility outside of my research. And I still take some classes. There's journal clubs that we do with grad students together just to learn more about a subject of interest. And those are good.
That's really interesting, actually. I never did journal club, but that sounds like a really kind of cool thing. Is it like a group session and you guys share stuff?
Yeah, so we go over a paper each week. Actually, I was in a very untraditional journal club for my first one, which was the professor. He's just this kind of crazy guy. He's very smart, and also he just takes a different approach to things. So what he had us do was pick a topic in the cancer field and read all of the papers about it from the very conception of this topic to present, and present that in a 45-minute presentation to the class.
Once a week?
An insane. It's once a week, but people sign up for one slot, so I only had to do it once. But it fell during a time where I had to give my own lab meeting presentation. I was busy teaching. I literally did not sleep. My NK cells were very unhappy. But yeah, it was very good because I enjoyed watching other people's presentations, and I learned a lot about the field that I picked, which was actually about how different gut microbes can actually cause cancer, such as Helicobacter pylori, which was historically found to cause colorectal cancers and other stomach-related cancers, and it was considered a big risk factor. There's obviously other risk factors, but that was one of them, and that's why Helicobacter is very bad if you get it. But yeah, anyways,
that's really cool.
Most of the time, yeah.
I'll take notes of that if I end up going into academia. That's something I could consider organizing for myself with my own students in the future. That'd be really cool.
No, it's great. Go. Anyways.
Sorry, I was going to go on to the next question. Tassia was asking, curious how easy it is to find a job in your field. Now, this question, I'm just going to jump in to say that you haven't been looking for a job yet because you're still a student. So I guess you don't have experience. But if you know of anything that you'd like to add, you're more than welcome to answer.
So I think it really depends. So my field is, again, like more biotech immunotherapy related. If I was trying to become a faculty member, so a professor, it would be quite difficult. I believe the available job positions are less than 10% or something like that. It's very low. So I'm not interested in becoming a faculty member, not because it's difficult, but because I personally don't want to stay in academia. If biotech right now, especially in where I'm at, so I'm on the West Coast of the US, it's not a good market currently because a lot of big biotech firms have been scaling down just due to like lack of funding, and maybe things are not working out so well. It's not a great market right now, but yeah, I would say that there's like some colleagues of mine who graduated recently, they got a job very quickly. It probably has to do with like some of the reputations of their labs. My advisor is very on it with forming collaborations. So that's why right now I'm working with a biotech company, testing their drugs to kind of build a relationship. And so that might serve me later on when I'm looking for a job. I can just sort of pool on that network. So that's, again, an important facet when you're looking for a graduate student advisor, is to see what is that advisor's outlook on industry. If they're very against it, you might have a very hard time finding support and a job when you're done. And so that was another big reason why I picked my advisor, because he was very much pro industry and supportive of whatever it was that I wanted to do.
Yeah. You're absolutely right about what you said about the whole competition about academia. Like, I don't think a lot of people realize, you know, just how tough it is out there, because let's say you have your supervisor. How many postdocs do they have and throughout their life? How many PhD students they produce? Ultimately, the way you should visualize it is, out of every person who ever is in their research group, only one person will replace their job, you know, ultimately in the end.
Exactly.
So I'm just looking at my own research group. My lecturer, he produced at least 20 students already, including postdocs and PhD students. And so already I'm competing at a 5% level. And just to put that number in perspective, that is just, that you just can't compete with that. Even if you do compete with that, like, what do you do with the other 95%? So I'm very encouraging people to not like tunnel vision on academia. There's other things out there, and there's a reason why you do PhD for other things out there. Taking out the competition is not a valid thing to do, Spooky One. Don't be giving people ideas.
Whoa.
Speaking of Spooky One, I did skim over his question earlier because it was I thought it was a bit of a joke. So he's rephrased the question so it's not too jokester. In my defense, Spooky One always asks meme questions, so I'm very hesitant to ever share anything.
Okay, Spooky
so
One.
The question is Spooky had sepsis before. Was that an issue with T cells attacking normal cells? What can make them misidentify a target? Do you know much about sepsis?
So I'm not studying sepsis actively, but it is not a T cell issue. So sepsis is when bacteria enter your bloodstream, and this can usually happen because of some other issue. For example, like let's say the bacteria enter the bloodstream through some leakiness in your gut barrier, or through some infection in the skin, like necrotizing fasciitis or something like that. So it's sort of a secondary thing that happens, although I'm sure there's other ways as well. But it's just essentially an issue of your immune system being unable to deal with this rapidly replicating onslaught of bacteria in the bloodstream. And then what happens is you do get some of these immune cells, like neutrophils and things, that do respond very quickly to the bacteria, but they're going to start producing a lot of inflammatory cytokines. This is going to start kind of creating a lot of this like increase of blood flow, and it can actually increase body temperature and then actually cause some organ damage and stuff like that. It can just spiral out of control very quickly. So yeah, but other than that, I don't know too much about sepsis. It is not fun, and it's incredibly, it's not necessary. Yeah, it's not T cells recognizing the wrong thing.
Okay. Okay. Thank you for explaining that.
Yeah.
Tetsu asks, if you do, how do you communicate the science of the therapies to doctors, physicians? And are there any challenges? To context, Tetsu's a young doctor as well, MD himself. So awesome. He's asking from a point of experience.
Oh, I apologize for my pre-med jab earlier. So I actually don't interface with doctors. However, what I did prior to graduate school was I was working in the brain tumor immunotherapy field, and my advisor there, so I was just a lowly lab manager / research associate, and COVID happened. And so we all had to go home. Well, not everyone, but I was sick actually at the time, and out of an abundance of caution, I was told to stay home for 4 months, and I was bored out of my mind. And so what I did was, my advisor had always got a lot of requests for research reviews. These research reviews are on the current status of immunotherapy for, in this case, glioblastoma. And he wanted me to write some, or I asked if I could write it. So I wrote 2 research reviews on the current status of the immunotherapy for this field. And it was supposed to be written in a way where physicians can read it very quickly quickly, and then just take the important bits and then apply it in their own practice. And I actually collaborated with several neurosurgeons at the time, and they helped with writing and just like feedback on these reviews. So that was the most interfacing that I had with actual oncologists and neurosurgeons who treat brain tumors. And it was a very cool experience, and actually something else I recommend for people who are trying to get into graduate school and maybe you just don't know what you want to do yet. If you're already working in an academic lab, ask your advisor if you can write a review, because they always get requests for reviews. And it's a great way for you to become familiar with the field, get your name on a published paper, and have something to kind of talk about and show when you're going through the graduate school application process.
Okay.
Yeah.
Yeah. Thank you so much for throwing in the advice. I'm sure there's a lot of people. We do have a lot of members of the community who are young and still looking to go into postgraduate schools, postgraduate programs. Another question, and I will say that this is the final question from you guys because I want to ask my own personal questions. What would field work look like for you, as in applying your methods? So I kind of asked that earlier in the end, but if you want to rephrase it again.
So field work, I guess it would not necessarily be like field work as if I was a geologist, for example, going out and studying different specimens like that. Field work. So I think the trajectory that I envision for myself and what a lot of people end up doing with a PhD in something like immunology is they go and work typically in pharma to help develop immunotherapies. And there are some positions in pharma where you cross over and actually collaborate directly with the clinics, so hospitals, and with bench scientists. And actually something I would like to do is kind of have this sort of coordination-like role. I don't want to be a clinical coordinator, but I would like to be someone who is kind of communicating with the labs doing the preclinical studies in mice and testing the drug or therapy, whatever it may be, and then making sure that that's up to snuff with the FDA standards, and then going over to the industry that's interested in turning this into an actual clinical trial and testing it into a product, and kind of making sure that all of those pieces sort of align, and that way we can get drugs from the bench into a patient much faster, because typically this is so crazy, but from the point of discovery to the point where a drug actually makes it into a human, that can take 10 to 20 years. It's a ridiculously long process. So you might read about a really cool discovery that some immunologist makes today, and that might not be applied in a human until 20 years from now. It's very long. So I want to help speed that up.
Well, that's still better than nuclear fusion science. That's my physics bone to pick with, because it's always 10 plus years. But I understand why it takes so long in anything to do with the body, human testing, anything like that.
Because you don't want accidents to happen.
No, we don't want a biohazard or a Resident Evil situation happening. But not that I'm claiming that, biologists are planning on making zombies. That's a completely bogus statement, of course. Totally not going to happen.
Yeah. And I know, like, big pharma, the dark side is strong. I would press back slightly. I do understand that a lot of pharma is driven obviously by profit, but I think in terms of getting practical solutions for patients, that is the main sort of trajectory that you have to go through.
Ultimately, it's
and
money,
maybe I'm
it's funding. You need it.
Yeah, you need money to do these experiments. It's so expensive to just, do mouse studies, for example. I am like the amount of money we could pay for antibodies, and we use so many of them like every year. It's like thousands and thousands of dollars. We easily spend over $1 million just on our own basic research alone. So having collaborations and also working in pharma is the way to get the money to do this research so that we can keep the field moving forward. And I know that there's a lot of dark sides of pharma, and I'm just maybe an idealistic person where I try to be like, but there's also good things. But yeah, no, I recognize that it's good and bad. But anyways.
Of course. I just, in my slight defense, that it was only a playful jab. But I digress. So I'm going to jump in with my questions. So a couple of things. These are kind of three fun questions coming up to the end. As a scientist, clearly working in a biology setting lab, Is there any movie or sci-fi TV show that you've thought the science or the practice has actually not been that terrible? I know CSI, like I'm not a person who works in that kind of lab, but I cringe at CSI scenes. However, is there anything that you've ever watched and you're like, you know what, that wasn't actually half bad?
You know, I should have known this question was coming. I don't actually have anything off the top of my head.
How about something
I don't watch
atrocious and terrible that made you want to burn your eyes out?
I do always, so I can't think of anything specifically, but I do hate the times when people are holding the pipettes upwards so the liquid can just fall back into the filter, they're not using gloves. Yeah. If you look at stock photos of scientists, you'll see a lot of really cringeworthy things where people are like holding the pipette without a tip on it and then like the little tube, and I'm like, what are you pipetting? There's no tip. There's some really bad stuff out there. But yeah, no, I can't. Specific example. Oh, mouth pipetting. Oh, this is actually a fun story. I used to mouth pipette when I worked in a plant pathology lab a long time ago. And so we didn't have a lot of funding. So we did things a lot of the old archaic way. And I had this little aspirator tube in which I would suck up the insects, which we would use for transmitting our virus onto a plant. And there's a little piece of cloth that's supposed to filter and keep the insect from going into your mouth that is in the tube. But somebody in our lab had broken it. They had not replaced it. And I just go grab it, start sucking up the insects, and realize none of them are going into my tube, and they were all in my mouth. And that was I actually still kept mouth pipetting from that point on, but I always checked my cloth to make sure it was secure. But yeah, that was a fun time.
Oh my days. No, that sounds like, yeah, you learn from experience. I hate to admit that I did something very similar, not with insects, of course, because of course I wouldn't do that.
Was it worse?
But I was working, obviously batteries, obviously acids, and I needed to get a flow going, and basic physics, raising the pipe to a certain level, but you also need to start the flow for the flow to start happening. And how do you start a flow? You suck on something. Just and suck. Yeah, so do be careful about that in future. Acids taste funny. Moving on. So a little bit more about yourself. Is your username is slightly caffeinated? Is there any story behind your username?
So I love caffeine. I specifically love coffee. So my stream typically revolves around me making a espresso before I start. And I got into making espresso at home about 5 years ago. And I used this like lever press machine. Mm-hmm. And at the time, it was a pretty novel device. So I started an Instagram and called myself Slightly Caffeinated because all the other variations of caffeinated were taken. I'm actually friends now with the person whose username I wanted. Their name is Mostly Caffeinated. So we have a little bit of a vendetta going on where I'm like, I want your username. But I am Slightly Caffeinated, and I love making good coffee. And I really got into this because making espresso is very similar to performing an experiment. You do a lot of weighing, you have a lot of variables to control for it. When I first got into espresso, I had an Excel sheet where I listed every single variable and I would change them and try to find the optimal setting for each of the types of beans that I have.
Damn.
Yeah, I love it. I love it a lot. And I always try to force my love of coffee onto other people, such as my Twitch chat.
Do you have this on your Twitch chat? Your Excel sheet?
Yeah.
Oh, class.
I don't have the Excel sheet.
Okay,
no, I don't. Yeah. I could. That's actually a good idea.
It could be a command. You could share it if you want to. Of course, if you want to publish the data, you might want to protect it, of course, for science.
Oh, yeah. It's not publishable. One of the biggest things with coffee is that, unfortunately, some of the variables you can't control well. And so I realized this, this Excel sheet isn't perfect. This method is not perfect. So at some point, I just do the process because I love it and then enjoy whatever I get out at the end.
No, I love the scientific application of trying to understand and discover what the truest, bestest form of coffee is out there. I appreciate the dedication. Oh, and then that leads finally to my last question. And it's not really a question, but it's kind of a nice conclusive question to summarize the whole stream, because ultimately we are giving a huge shout out in the title, Slightly Caffeinated. Make sure to give her a follow if you don't already. But the question is, to promote your stream, what is it that you do on Twitch? that in the wee hours of the night when I'm asleep?
So I do a variety of things. I do currently gaming about 50% of the time, and science chats the other 50% of the time. Obviously, coffee is woven in there. I will typically stream on Saturdays, for example. Those are my fun days. So I'll make a fun coffee drink, espresso based. I'll maybe do a little chat about the coffee, and then I play, mostly FPS games, which I'm terrible at. It's mostly been FPS games. So like Helldivers,
Nice.
Hell Let Loose, lots of Hell. What else? Rust. I used to play Rust a lot, but I haven't played that in a while. And then for science chats, I like to do a little recap of the news sometimes. This is a little bit more sporadic because it depends on how busy I have been during the week and how much preparation time I have.
Yeah.
But I do try to collect some interesting microbiology, immunology related news, go over that with chat, and then I like to pick special topics. So in my Discord, I have a topic suggestion channel where people can put topics that are related to the immune system, cancer, or microbiology, and I will at some point try to cover that in a lengthy detail in stream, and then we can chat about it. And that's usually on Monday nights, although today or this week it's going to be Wednesday because yesterday was just so stressful. But yeah, so yeah, that's the other thing. My schedule is kind of a little all over the place at times because of the demands of grad school, and sometimes I'm just not able to have the energy to go live, but I do love it, and I do love my community, and yeah.
Absolutely. No, thank you so much for making time out of your busy schedule to do this Tofu Tech Talk. I really, really appreciate it.
Of course.
I think Slightly Caffeinated absolutely deserves a big round of applause in the chat, and by that I mean mute ones, fake clapping in the chat. It's been a complete honor to have you as a guest. I think as an interviewee or as a victim, depending on what phrasing you want to use, you were absolutely, absolutely phenomenal. Really, really great. I think the way you answered questions was absolutely fantastic. Only good compliments and good statements from my end. So yeah, I really appreciate you coming on the show, taking time out of your day to, I'm sure you might have been nervous. Oh, you were a little nervous. You were talking about the nightmare.
I was a little nervous. I had a nightmare that I was 2 hours late.
Exactly. I'm so sorry that you were nervous. I hope.
No, it's good to be nervous because that means you're more prepared a little bit. And I always get a little bit nervous. It's just normal. But I had a lovely time. I really enjoyed chatting with you and your community. I love your Tofu Talk ideas. Like, this is such a fabulous stream. I love catching them. And it's just been an honor to be on one of your Tofu Talk talks. It's great. It's a dream.
No, you're too sweet. I'm glad you're caffeinated. Exactly. I think everyone is glad that you're caffeinated. I love the coffee theme of your stream, and it's into your username. That's really, really good. So I will say again, guys, if you don't follow Slightly Caffeinated, make sure you do. Shooter games, talking about coffee, and talking about science. What more could you want in a Twitch stream? Honestly.
Thank you.
Do you have any last statements, any last words? Sounds very threatening.
Oh goodness. I just stay caffeinated. If you have any questions at all, feel free to pop into my Discord about, we can chat about cancer, immunology, that type of stuff. I'm always
the title?
Yes. Yeah. Yes.
Yeah,
the link is in the title.
Just hover over the name and the Discord link is there.
Yeah, get good sleep, take care of your body, don't be stressed, take care of your natural killer cells, and drink coffee, which
is that bad? It's a good thing. Is coffee good for
so for natural killer cells, we don't actually know, but in general, people who drink coffee, they actually have longer lives than people who don't. So, but that's retrospective data. we can discuss this in more detail later, but yeah.
All right. Thank you again so much. Really appreciate it. Thank you so much, Lab Rats, for the hype train. You little shits. I know you guys did that on purpose, but thank you so much for that hype train and taking us to Isengard before we start a stream. I'll see you guys all tomorrow for some special theory of relativity. But again, goodbye, Slightly Caffeinated. Thank you again so much.
Goodbye. Thank you so much for your time. All right, I will hang up now.